Everyone, and welcome back to my channel. So for viewers who are new to my channel, I'll just give a brief introduction. So I am Triya and I generally post videos and lectures on tissue engineering, regenerative medicine and its associated topics. I have previously posted lectures on the basics of tissue engineering and about the integral components of it, which includes the cells, the scaffold, and other such components. And now I have also started to post about other major things that are involved there, which is the cell and the ECM interactions. So I've already posted another video, which is in the part 1 video. So people should also look into that. So this is a continuation of that part. And with this, we'll start today's class. So the extracellular matrix or ECM is an intricate network that is composed of an array of multi-domain macromolecules that is organized in a cell or tissue specific manner. So the components of the ECM, they link together to form a structurally stable composite that contributes to the mechanical properties of the tissues. Basically, the ECM is also a reservoir of growth factors and bioactive molecules. It is a highly dynamic entity that is of extreme vital importance, which determines and controls the most fundamental behaviors and characteristics of cells, such as the proliferation of cells, adhesion, migration, polarity, differentiation, and apoptosis. So, the cells are able to communicate between themselves with the help of ECM, and ECM also affects the cell shape. So, the orientation of cytoskeleton in cell orients the assembly of secreted extracellular matrix molecules in the vicinity. The oriented extracellular matrix thereby reaches the cells and orients the cytoskeleton of those cells. Further, the cells and they now secrete an oriented matrix in their vicinity. So basically, in this way, they order the cytoskeleton and it is propagated to the cells. Further, as I've already mentioned, that the ECM, it affects the cell survival and proliferation. So the attachment of cells to ECM is essential to support cell survival, growth, and proliferation. And the lack of these interactions can trigger a type of cell death, which is also certainly is known as the apoptosis or anoikis. So there are several studies that point out that if there are certain alterations of the ECM composition, then it indicates that they are often responsible for several pathological conditions such as cancer, of which it has been demonstrated to be occasionally the main promoter. ECM does not always represent a pro-survival stimulus. Among the different array of ECM molecules, a set of proteins can negatively affect the cell viability and are thought to play an important role in tumor progression. For this reason, attention has been focused on these molecules as potential tools or targets for therapy. So, the cell also dies by apoptosis, and there are certain cases where the cell spreads, survives, and grows. So, everything happens and there is a sequential balance between it so that proper healthy state and a condition is maintained. So ECM is also responsible for promoting the cell survival, proliferation. ECM molecules connect to the cells through several molecules like the integrins, syntegens, and other receptors, among which integrins are the heterodimeric receptors that are composed of alpha and beta subunits. In vertebrates, the family encompasses 18 alpha and 8 beta subunits that can assemble into 24 different integrins. Both alpha and beta subunits are transmembrane proteins with large modular extracellular domains and single transmembrane helices and short cytoplasmic regions that can mediate the cytoskeleton interactions. So the integrins can be grouped into subgroups based on ligand binding properties or based on their subunit composition. Further, major matrix binding integrins are the beta 1 integrins with affinity to fibronectin, collagen, and laminin. Integrins basically function as links between ECM and the cytoskeleton. Further, we can also describe it as that integrins are the transmembrane receptors that facilitate the cell-to-cell and the cell-to-ECM adhesion. Upon ligand binding, the integrins activate the signal transduction pathways that mediate cellular signals such as regulation of the cell cycle, organization of the intracellular cytoskeleton, and movement of new receptors to the cell membrane. The presence of integrins allow rapid and flexible responses to prevent to events at the cell surface. For example, the signal platelets to initiate an interaction with the coagulation factors. Further, several types of integrins exist, and one cell generally has multiple different types on its surface. Integrins are found in all animals, while integrin-like receptors are found only in plant cells. Integrins, they work alongside other proteins such as the cadherins, the immunoglobulin superfamily, cell adhesion molecules, selectins, and syndicins. So 20 nanometer above the cell surface, they are dependent upon the calcium and magnesium. Integrins couple to ECM to the cytoskeleton through several cytosolic proteins like the tallin, alpha-adenine, paxillin, filamin, and vinculin. A living cell is constantly interacting with its environment to strive, and thus the cell surface receptors play a vital role in mediating these interactions. Integrins are the major metazoan receptors. They are alpha-beta heterodimeric transmembrane molecules, and each of the subunits consists of a very large extracellular domain, a single transmembrane segment, and a short cytoplasmic tail. By regulating the cell to the ECM contact, cell-cell adhesion, and cell-pathogen interaction, integrins take part in a wide range of biological processes, including the development, angiogenesis, immune response, cancer, hemostasis, and others. The name of integrin inherently means to integrate the extracellular and intracellular environment, and thus it got its name. The most important aspect of their function is the bidirectional signaling that takes place across the plasma membrane. On one hand, the ligand binding to the integrin extracellular domain is transduced to the cytoplasm in the classical outside-in direction to regulate intracellular activities. On the other hand, integrins are often expressed on the cell surface in a default low affinity ligand binding conformation. When cell becomes activated, for example by cytokine, integrins are rapidly activated within the cytoplasm. They undergo large conformational changes, resulting in a dramatic affinity increase for ligand binding. The process is commonly termed as the inside-outside signaling. So the inside-out signaling of integrins regulates the ligand binding affinity of the cell surface receptors in response to changes in the environment for the cell survival, as it can be seen in the diagram. So the specific binding of the cytoplasmic tail of the integrins beta subunit by the intracellular protein tallin is a key step of the inside-out signaling. A pull-push mechanism is there which has been proposed to explain how the PIP2 enriched membrane disrupts the dual autoinhibition of the N-terminal tallin firm domain by the C-terminal tannin rod domain such that activated tannin firm can reach the beta 10 for integrin activation. So while the alpha subunit of integrin seems to be more responsible for the extracellular ligand binding specificity in the outside in event, the short cytoplasmic tail of the beta subunit is the region where there is interaction with the regulatory proteins in the inside-out process. So when the integrins are in the resting state, the alpha and beta cytoplasmic tails are in close proximity, which constrains the integrin in a low affinity conformation. Further, the dissociation of the two tails by signals within the cell subsequently triggers conformational changes, thereby activating the integrins. In this, we'll move on towards discussing more details about the cell-to-cell and the cell-to-matrix adhesions. Cells combine to form tissues, and this requires the cells to adhere to one another in order to form a functional unit. So cell adhesion is the process by which the cells interact and attach to neighboring cells through specialized molecules of the cell surface. This process can occur either through the direct contact between the cell surfaces, such as the cell junctions, or indirect interaction, where cells attach to surrounding extracellular matrix, which is a gel-like structure that contains molecules released by cells into the spaces between them. Cells adhesion occurs from the interactions between cell adhesion molecules, which is known as CAMs, and the transmembrane proteins that are located on the cell surface. Cell adhesion links the cells in different ways and can be involved in signal transduction for cells to detect and respond to changes in the surroundings. Other cellular processes regulated by cell adhesion includes the cell migration and tissue development in multicellular organisms. Alterations in the cell adhesion can also disrupt important cellular processes and lead to a variety of diseases that includes the arthritis, cancer. Cell adhesion is also essential for infectious organisms such as bacteria or virus to cause diseases. So there can be majorly two types of adhesion, which is the cell-to-cell adhesion, and the other is the cell to the extracellular matrix adhesion. So we'll go into more details in the further next slides. Coming to the brief description about the adhesions. So talking about the cell-to-cell adhesions, which is brought about by the cell adhesion molecules, which are also known as CAMs. So CAMs are classified into four major families, which are the integrins, the immunoglobulin, which is the Ig superfamily, the cadherins, and the selectins. Further, the cadherins and the IGSF are the homophilic CAMs, as they directly bind to the same type of CAM on another cell, while integrins and selectins are the heterophilic CAMs that bind to different types of other cell adhesion molecules. Each of these adhesion molecules has a different function and it recognizes different ligands. Defects in the cell adhesion are usually attributable to defects in the expression of the CAMs. In multicellular organisms, bindings between CAMs allows cells to adhere to one another and also create structures which are known as the cell junctions. According to their functions, the cell junctions can be classified as the anchoring junctions, which is the adherens junction, the desmosomes, and the hemidesmosomes, which maintains the cells together and strengthens the contact between the cells. Occluding junctions are also known as the tight junctions, which seal gaps between the cells through cell-to-cell contact, making an impermeable barrier for diffusion. Further, the channel-forming junctions, which are the gap junctions that links the cytoplasm of adjacent cells, allowing transport of molecules to occur between the cells. Signal relaying junctions, which can be synapses in the nervous system. Further, talking about the cell-to-matrix adhesion, it is brought about by the adhesion receptors between the ECM and which interacts with the adapter elements of the cell. Further, we'll also talk into details about the cell-to-cell interactions. Further, as I've already mentioned previously, that there are different types of interactions that takes place between the adhesion proteins, which can be grouped into four major types. So first, it can be homophilic. So homophilic is the one where the adhesion is created by interaction between two similar adhesion molecules. Another type can be the heterophilic type, where the adhesion can be created by interaction between two different adhesion molecules or between adhesion molecules and cytoskeleton or extracellular matrix proteins. Further, it can also be of homotypic type, which is the adhesion between similar molecules, and heterotypic, as the name suggests, is the adhesion between different molecules. So the cell adhesion molecules are a subset of cell surface proteins, as I have already mentioned. So those are involved in the binding of cells with other cells or with the extracellular matrix in a process called cell adhesion. In essence, the CAMs help the cells to stick to each other and to their surroundings. CAMs are crucial components in maintaining the tissue structure and the function. In fully developed animals, these molecules play an integral role in generating force and movement, and consequently ensures that organs are able to execute their functions normally. Further, in addition to serving as the molecular glue, CAMs play important roles in the cellular mechanisms of growth, contact inhibition, and also apoptosis. Further, aberrant expression of the CAMs may result in wide range of pathologies that can range from frostbite to cancer. There are major 5 classes of the CAMs or the cell adhesion molecules, which I'll be discussing in the next slides. So there are the immunoglobulin superfamily of cell adhesion molecule, then cadherin, integrin, and the superfamily of C-type of lectin-like proteins. Proteoglycans sometimes are also considered to be class of the CAM. Further, talking about the cell-to-cell adhesion molecules, first we'll be talking about the calcium-dependent adhesion molecule, which is known as the cadherins. So, cadherins are the transmembrane protein that mediates the cell-to-cell adhesion in animals. So, it is evolutionarily very ancient and it is widely expressed over 12 different known types, and almost all vertebrate cells express one or more of these cadherins. So talking about its structure, so it is a single pass transmembrane glycoprotein that is composed of around 700 to 750 residues. So it regulates the contact formation and stability, and cadherins also play a crucial role in tissue morphogenesis and homeostasis. So talking about its functions, so there are major 3 functions of cadherins in cell-to-cell contact formation and stability. 2 of those functions lead to decrease in interfacial tension at the forming cell-to-cell contact, and thereby they promote contact expansion. First, by providing adhesion tension that lowers the interfacial tension at the cell-cell contact, and second, by signaling to the actomyosin cytoskeleton in order to reduce the cortex tension and thus the interfacial tension at the contact. The third function of cadherins in cell-to-cell contact formation is to stabilize the contact by resisting the mechanical forces that pull on the contact. So coming to the types of binding, so it can be of different types. It can either be the interactive with the actin cytoskeleton, which is brought about by the cadherins N, P, R, B, and E. Others include the desmosome associated, which is the desmoglins and the desmocolins, and further the photocadherins. About the second type, which is the immunoglobulin superfamily or the Ig superfamily. So these are the non-calcium dependent adhesion molecules, and the immunoglobulin superfamily is a large protein superfamily of cell surface and soluble proteins that are involved in the recognition, binding, or adhesion processes of the cells. Molecules are categorized as members of the superfamily that is based on shared structural features with immunoglobulins, which are also known as the antibodies. They all possess a domain which is known as the immunoglobulin domain or the fold. Members of the immunoglobulin superfamily include cell surface antigen receptors, co-receptors, and co-stimulatory molecules of the immune system. They also contain molecules that are involved in the antigen presentation to lymphocytes, the cell adhesion molecules, certain cytokine receptors, and intracellular muscle proteins. They are commonly associated with the roles in the immune system. Otherwise, the sperm-specific protein, a member of an immunoglobulin superfamily, has also been identified as the only sperm membrane protein that is essential for the sperm and the egg fusion. So there are certain proteins of the immunoglobulin superfamily that possess a structural domain which is known as an immunoglobulin domain. The Ig domain are named after the immunoglobulin molecules, and they contain about 70 to 110 amino acids and are categorized according to their size and function. Further, the Ig domains possess a characteristic Ig fold which has a sandwich-like structure formed by two sheets of antiparallel beta strands. Further, the NCAMs, which is the non-calcium dependent adhesion molecules, or the Ig superfamily, nerve adhesion molecules, ICAMs and L1. And talking about these are also evolutionary, ancient, and widely expressed. Coming to its structure, so it is a single-pass transmembrane protein that may bind to the cytoskeleton inside the cells. Further, talking about the types of adhesion, so they can have both homophilic and heterophilic interactions. The homo-to-neural specific Ig cell adhesion molecules and the heterosystemic Ig CAMs. Talking about its major functions, so it helps in the neurite outgrowth, myelination, and also the firm adhesion of the leukocytes. Talking about the next type, which is the selectin. So the selectins are a family of cell adhesion molecules or CAMs, and all selectins are single-chain transmembrane glycoproteins that share similar properties to C-type lectins due to a related amino terminus and calcium-dependent binding. Selectins tend to bind to sugar moieties and so are considered to be a type of lectin. Further, the cell adhesion proteins that bind sugar polymers. All the three known members of the selectin family, which is the L, E, and P selectin, share similar cassette structure, which is an N-terminal calcium-dependent lectin domain and an epidermal growth factor-like domain, along with a variable number of consensus repeat units. It also comprises of a transmembrane domain and an intracellular cytoplasmic tail. The transmembrane and cytoplasmic parts are not conserved across the selectins, and they are being responsible for targeting to different compartments. Though they share common elements, their tissue distribution and binding kinetics are quite different, reflecting their divergent roles in various pathophysiological processes. Further, they are expressed only in vertebrates in circulating or circulatory cells, which are the endothelium and the blood cells. They are the transient transmembrane binding proteins, which are known as the lectins, and in the presence of calcium, they bind to specific oligosaccharides on another cell. And as already mentioned, its structure is very unique as it is a single transmembrane polypeptide. So, selectin, as already mentioned, are involved in constitutive lymphocyte homing and in chronic and acute inflammation processes, which includes the post-ischemic inflammation in cell, muscle, kidney, heart, skin inflammation, atherosclerosis, glomerulonephritis, and lupus erythematosus, and also cancer metastasis. So this image shows the structure where you can see the catenaries, the adhesion belt, the lumen, and the actin filaments that are inside the microvillus. Further, there are also microvilli that extends from the apical surface. And as the diagram suggests, that there are also tight junctions that are being found along the side of the bundle of the actin filaments. So there are lateral plasma members of adjacent epithelial cells and the basal surfaces. Further, talking about the second major interaction, which is the cell to the ECM adhesion. So now we'll be talking about how the cell adheres or interacts and how it attaches to the extracellular matrix and how it takes place. The linkage of the extracellular matrix to the cell, it requires transmembrane cell adhesion proteins that can act as matrix receptors, and they tie the matrix to the cell cytoskeleton. Although we have seen that some transmembrane proteoglycans function as co-receptors for matrix components, the principal receptors on animal cells for binding those most extracellular proteins, including the collagen, fibronectin, laminin, are the integrins. So they are a family of transmembrane adhesion molecules, usually the glycoproteins, that exist in variable activation states. Extracellular matrix receptors on integrins have selective affinity for certain matrix proteins that allows the cells to explore their environment. Further, they have an alpha and a beta subunit, which rises because of the alternating splicing has led to 16 different alpha chains and 8 different beta chains. The main function can be the white blood cell binding to the endothelium. So the integrins are crucially important because they are the main receptor proteins that cells use to both bind to and respond to the extracellular matrix. An integrin molecule is composed of two non-covalently associated transmembrane glycoprotein subunits, which are known as the alpha and the beta. Further, because the same integrin molecule in different cell types can have different ligand binding specificities, it seems that the additional cell type specific factors can interact with integrins to modulate their binding activity. Further, we'll move on towards the next part, which is the cell junctions in the tissues. Further, the epithelium is a type of body tissue that forms the covering on all the internal and the external surfaces of our body, and also lines the body cavities and hollow organs, and is a major tissue in the glands. So the organs in our body are composed of four basic types of tissue, which includes the epithelial, connective, muscular, and nervous. So coming to epithelial tissues, and why do they need adhesion molecules for? So basically, because there are certain junctions that are required for maintaining the proper cell integrity, and those junctions are the tight junctions, the adherence junctions, the desmosome, and the hemidesmosome. So here in this image, it shows the structure and organization of different junctions and microvillus, and it also demonstrates the gap junction, connective tissue, several types of junctions that are present in between the cells and are properly maintained and are required for maintaining proper functioning of the cell. Coming to the tight junctions. So tight junctions are the intercellular adhesion complexes in epithelia and endothelia that control paracellular permeability. So this paracellular diffusion barrier is semi-permeable. So it is size and charge selective. Further, the paracellular iron permeability at tight junctions is largely determined by their claudin composition. So before going into claudins, so the basic protein composition of tight junctions include the occludin, the claudin, and the junction adhesion molecules, which are also known as JAMs. So claudins are a family of transmembrane proteins that are thought to form gated ion selective paracellular pores through the paracellular diffusion barrier. Further, the tight junctions form the border between the apical and the basolateral cell surface domains in polarized epithelia. And they also support the maintenance of cell polarity by restricting the intermixing of apical and the basolateral transmembrane components. Further, tight junctions are an integral component of the evolutionary conserved signaling mechanisms that control epithelial to cell polarization and the formation of morphologically and functionally distinct apical domains. Further, the tight junctions form bidirectional signaling platforms that receive signals from the cell interior, which also regulate their assembly and function, and that transduce the signals to the cell interior to control the cell proliferation, migration, differentiation, and also survival. Further, tight junctions are part of an interconnected network of adhesion complexes that also includes the adherence junctions and focal adhesions. So basically, these adhesion complexes crosstalk through direct protein to protein interactions as well as by transmitting these signals to each other that also influence their assembly and their function. Further, in this slide, as the image is visible, so this is a graphical representation of the tight junction and how it is sealed so tightly by the different proteins, which are the claudins, the occlutin, and the JAMs. Further, talking about the other junctions, which are the adherence junction, which is the adhesion belt that attach to actin. Further, desmosomes help to attach to the intermediate filaments. Similarly, the focal adhesions and the hemidesmosomes. So, talking about the adherence junctions. So, adherence junctions are the cell-to-cell adhesion complexes that are continuously assembled and disassembled, thereby allowing cells within a tissue to respond to several forces, which can be the biochemical signals, and also there are structural changes in their microenvironment. So the events leads up to the adherence junction formation are still not entirely clear, but they ultimately result in the recruitment of the transmembrane catenins and also the cytoskeletal adapter proteins. Further, going into more details about the adherence junctions, so the major composition is the cadherens, and the function is it can contract with the help of myosin. There is folding of sheets into tubes during the morphogenesis and other folding processes during the morphogenesis. And the major binding partners are the catenins and via catenins to the cytoskeleton, which is the actin. So the exact formation and function of the adherence junction can be described over defined steps, including the initiation, cadherin recruitment, and recruitment of the plaque proteins. So adherence junction assembly occurs in a variety of contexts, such as during development, when migrating cells first encounter each other to form a tissue, as well as in pre-existing tissues when cells divide, change their shape, or move in response to biochemical or mechanical stimuli. A large body of work and several researchers have investigated that the adherence junction initiation during embryonic morphogenesis and in cell culture when migrating cells first contract each other. It is important to note that the adherence junction form and disassemble throughout life, not only during development, and this turnover is vital to the homeostasis of the epithelial tissues. Further, talking about the desmosomes, so desmosomes are the intercellular junctions of epithelia and cardiac muscle. They resist the mechanical stress because they adopt a strongly adhesive state in which they are said to be hyperadhesive and which distinguishes them from other intercellular junctions. Further, the desmosomes are specialized for strong adhesion and their failure can result in diseases of the skin and heart. They are also dynamic structures whose adhesiveness can switch between high and low affinity adhesive states during processes such as embryonic development and wound healing. The switching being signaled by protein kinase C. Desmosomes may also act as signaling centers, regulating the availability of signaling molecules and thereby participating in fundamental processes such as cell proliferation, differentiation, and morphogenesis. So this is all about the desmosomes, and to mention that they perform several functions, and the desmosomes structure have quite characteristic and electron dense structure. They are around less than 1 micron or 1 micrometer in diameter and consist of a central core region that spans the intercellular space between opposing cells and separates two identical cytoplasmic plaques that are associated with the intermediate filament network. It seems very likely that this unique, robust looking structure holds the key to understanding the basis of hyperadhesiveness. So, focal adhesions are the complex plasma membrane associated macromolecular assemblies that engage with the surrounding extracellular matrix via the integrin receptors and they physically connect with the actin cytoskeleton through the recruitment of numerous Fa associated proteins. Further, focal adhesions are the active sites for actin polymerization. In contrast to actin polymerization at the leading edge of the cell, actin polymerization at focal adhesions is strongly dependent on myosin II based or externally applied forces. Involvement of several actin nucleation or elongation factors in the actin polymerization at focal adhesions has been revealed. Among them, roles of several formins have been also extensively studied. So, focal adhesions can be described as the contact points for the cell with the extracellular matrix. These complex structures regulate the communication of the cell with the surrounding extracellular environment, and signaling through these focal adhesions regulates diverse cellular processes which includes the proliferation, migration, apoptosis, spreading, and differentiation. The principal components of the focal adhesions are the integrins, as already stated. So basically, they are the alpha beta heterodimers that regulate the cell matrix and cell-cell interactions. Further, focal adhesion kinase is a kinase that localizes the focal adhesions and they regulates the signaling in the focal adhesions. So this is all about the focal adhesions. Talking about certain examples, which includes the myotendinous junctions and the fibroblast migration in the connective tissue. In this slide, it shows the chart of the various junctions and its associated and designated function. So talking about the tight junction, so basically it acts as a sealing and it seals the neighboring cells together in an epithelial sheet to prevent leakage of molecules between them. Further, moving on towards the second junction, which is the adherence junction, which joins an actin bundle in one cell to a similar bundle in the neighboring cell. Further, desmosomes means and functions to join the intermediate filaments in one cell to those in a neighbor. Further, talking about the gap junction, it allows the passage of small water-soluble ions and molecules. Thereafter, talking about the last important junction, which is the hemidesmosome, it functions to anchor the intermediate filaments in a cell to the basal lamina. Further, this slide shows the different type of organization of cells in different organs. For example, in the muscle, epithelium, and the kidney glomerulus. Further, it becomes important to mention about the connective tissue. So, connective tissue are a group of tissues in the body that maintain the form of the body and its organs and also provides cohesion and internal support. The connective tissues include several types of fibrous tissue that vary only in their density and cellularity. as well as the more specialized and recognizable variants, which are the bone, ligaments, tendons, cartilage, and adipose tissue. Further, I'd like to mention that in the abdominal cavity, most organs are suspended from the abdominal wall by a membranous band which is known as the mesentery, which is supported by the connective tissue. Others are embedded in the adipose tissue, which is a form of connective tissue in which the cells are specialized for the synthesis and storage of energy-rich reserves of fat or lipid. The entire body is supported from within by a skeleton composed of bone, which is a type of connective tissue which is endowed with high and great resistance to stress owing to its highly ordered laminated structure and to its hardness, which results from deposition of mineral salts in its fibers, and the amorphous matrix. The individual bones of the skeleton are held firmly by ligaments and muscles and are attached to bone by tendons, both of which are examples of dense connective tissue in which many fiber bundles are associated in parallel array to provide great tensile strength. Further, we'll be discussing about the regulatory factors that are vital or which have an impact on the matrix synthesis, degradation, and function, and also they include the growth factors, cytokines, the hormones, vitamins, matrix metalloproteinases, and tissue inhibitors of matrix metalloproteinases. Talking about the vitamins, so a list of matrix regulators might historically start with the recognition in the 18th century that vitamin C was an essential nutrient that was required for hydroxylation of proline to hydroxyproline, and also its consequent stability of the collagen triple helix. And further, vitamin D plays an important role in the dynamic reactions of the matrix of bone. So coming to about the hormones, which are the estrogens and the glucocorticoids. So the effects of hormones on matrix production, stability, and degradation have been applied clinically for decades for a variety of conditions that ranges from estrogens for osteoporosis to glucocorticoids for inflammatory processes, which often have fibrotic sequelae. Growth factors or the cytokines may mediate some or many of these effects, while others may act directly on matrix components or their receptors. Talking about the matrix metalloproteinases, MMPs, so they are the zinc-containing enzymes that degrade most molecules of the ECM or the extracellular matrix. So matrix metalloproteinases are a family of enzymes that contain zinc, as I stated, in their active site and can degrade most of the matrix macromolecules found in the connective tissues. The activation of some of the MMPs is linked to integrin receptors and is promoted by SPARC, which is by SPARC. So MMPs are secreted by both matrix cells and the infiltrating leukocytes in response to the inflammatory mediators. Further, the MMPs are the major class of proteinases that are responsible for degradation of cartilage in the rheumatoid arthritis. Further, talking about the tissue inhibitors of metalloproteinases, which are also known as TIMP. So they are the zinc binding endopeptidases. So the TIMPs are natural inhibitors of the matrix metalloproteinases, collagenases, stromelisins, and gelatinases, and they are a group of zinc binding endopeptidases that are involved in the degradation of the extracellular matrix. So at least 3 different TIMs have been characterized. Each inhibits each are inhibiting, and all are known as all known in eukaryotic. So TIMs affect the activation of
pro-metalloproteinases, and they modulate proteolysis of extracellular matrix components. TIMs are particularly active in development during tissue remodeling and also in the pathologic disorders that are associated with the inflammatory processes and the tumor metastasis process. Because of this intimate relationship between the solid phase matrix components and the growth regulatory agents, TIMs play an important role in matrix cell regulation. Further talking about the growth factors, so TGF-beta plays a major role in matrix production and degradation. It also induces leukocyte margination and accumulation both through direct chemotaxis and by inducing cell surface integrin expression on the monocytes. As a consequence, TGF-beta promotes the monocyte adhesion to type 4 collagen, laminin, and fibronectin, which facilitates the cellular movement through the matrix. In that, TGF-beta also stimulates gelatinous or type 4 collagenase, and it plays a major role in inflammation and repair. Most growth factors directly or indirectly affect the matrix, which is a subject which is beyond the scope of this discussion. So proteoglycans are the molecules that contains carbohydrate structures which are known as glycosaminoglycans that are covalently attached to the protein core. Besides glycosaminoglycans, they usually contain oligosaccharides too. There are 7 different types of glycosaminoglycans that are found, and further, exceptionally, hyaluronan is always present as a free carbohydrate chain. Further, the 7 different types are the chondroitin, the dermatan sulfate, the keratin sulfate, heparin sulfate, and or heparin hyaluronan. So GAGs or the glycosaminoglycans are highly negatively charged molecules with extended conformation that imparts high viscosity to the solution. Further, GAGs are located primarily on the surface of cell in extracellular matrix, which is the ECM. Along with the high viscosity of GAGs comes low compressibility, which makes these molecules ideal for lubricating fluid in the joints. At the same time, this rigidity promotes the structural integrity to cells and provides passageways between the cells, thereby allowing for the cell migration. Further, talking about the common structural makeup of the GAGs attachment to proteins, proteoglycans or mucopolysaccharides. So, the biosynthesis of glycosaminoglycans is initiated. Of course, there are some exceptions, but majorly it is being initiated by adding the reducing size of xylose to the serine residue of the core protein. Sometimes also, threonine may accept the xylose. The reaction is catalyzed by a suitable enzyme, xylosyl transferase. Elongation of glycosaminoglycans is performed by the action of cells and the enzymes that aid one monosaccharide at a time to the end of the growing genes. So N-acetyl galactosamine transferase and glucuronide transferase are involved in the biosynthesis of chondroitin and dermatan sulfate. Sulfate is added simultaneously with carbohydrate chain polymerization, and inorganic sulfate is activated with ATP into phosphoryl phosphosulfate, and that donates the sulfate group to the glycosaminoglycans, and the donation is catalyzed by specific sulfotransferase that donate the sulfate into the carbon 4 or the 6 chain. The arrangement of sugar residues in GAG chains and the modification of specific sugars in the chains can determine their function and that of the proteoglycans containing them. Groupings of the modified sugars can control the binding of growth factors to certain receptors, the activities of proteins in the blood clotting cascade, and the activity of lipoprotein lipases. So the pentasaccharide sequences that are found in a subset of heparin GAGTs that control the activities of the anti-thrombinil and the inhibitor of the key blood clotting protease thrombin. When these sequences are sulfated at two specific positions, heparin can activate the AT3, which inhibits the clot formation. So, as it is visible in this chart, so the several GAGs, their localization and their functions or some comments on that. So talking about the hyaluronate, so it is localized in the synovial fluid, the vitreous humor, the ECM of loose connective tissue, and it acts as a shock absorbing molecules and they are large polymers. Talking about the second GAG, which is the chondroitin sulfate, so it is mainly localized in the cartilage, bone, and heart valves, and it is one of the most abundant GAGs. Further, the heparin sulfate is found in the basement membranes and also as components of cell surfaces, and it contains higher acetylated glucosamine than the heparin. Further, heparin is also another GAG which is component of the intracellular granules of mast cells that lines the arteries of the lungs, liver and skin. And it is more sulfated than the heparin sulfate. So if people ask you what is the difference between the heparin and the heparin sulfate, so apart from its localization, so heparin sulfate contains more acetylated glucosamine than heparin, whereas the heparin it contains more sulfated than the heparin sulfate. So talking about the dermatel sulfate, it is localized in the skin, blood vessels, and heart valves. Similarly, the keratin sulfate is also found in the cornea, bone, cartilage, which is aggregated with the chondroitin sulfate.
So from now, we'll talk about the several extracellular matrix proteins in the connective tissue. So the several proteins of ECM are the collagens, proteoglycans, adhesion proteins, hyaluronan and the elastic fibers. So thereafter we'll talk deeply and more clarity will be given to you about each of these proteins and how each of them are important in properly governing the function of the extracellular matrix and thereby helps to perform the functions of the cells.
So talking about the type 4 collagen, so like other collagens, it is composed of the repeating sequence containing the like glycine X and Y, where X is proline and Y is generally hydroxyproline. So the resulting molecule basically so it's a left-handed helix. First we'll talk about that, and so basically the N terminal and the C terminal it ends have the globular domains. So basically they are cleaved when secreted from the cell and they are in form of insoluble form. So in the ER and the Golgi they are glycosylated and the OH groups are added and SS links three chains. So N results into a triple helix. Further, the resulting molecule twists into an elongated left-handed helix, which is not an alpha helix, as you can see it in the image. So when synthesized, the N terminal and the C terminal of the polypeptide, they have globular domains, which keep the molecules soluble. As they pass through the endoplasmic reticulum and the Golgi apparatus, the molecules are glycosylated and the hydroxyl groups are added to the Y amino acid. And further also the sulfide bonds, they link three chains covalently. So the three molecules twist together to form a triple helix. In some collagens, for example type 2, the three molecules are identical, the product of a single gene. So in other collagens, example type 1, two polypeptides of one kind or gene product assemble with a second, quite similar polypeptide that is the product of a second gene. When the triple helix is secreted from the cell, usually by a fibroblast, the globular ends are cleaved off. The resulting linear insoluble molecules it assembles into collagen fibers. Further, they assemble into a staggered pattern that gives rise to the striations that are seen in the electron micrograph. Further, the type 4 collagens are an exception. They form a meshwork rather than the striated fibers. Talking about the good pastures syndrome, so some people develop antibodies against an epitope on their type 4 collagen molecules. They further then attach to the basal lamina of the epithelial cells and they fix the complement which damages the basal lamina. So good pastures syndrome is an example of an autoimmune disorder where the basal lamina of the lung epithelia and the glomeruli of the kidney are especially likely to be affected. So this is all about the assembly and the other details. Further, talking about the collagens and how it assembles. So, in the case of the interstitial collagens, the characteristic binding pattern is accounted by an ordered, staggered arrangement of the collagen molecules within the collagen fibrils and the collagen fibers. The manner of molecular packing within the fibril in turn is determined by the amino acid sequence. The way the molecules are staggered in the fibril, however, gives rise to the regions in which the molecules overlap and others in which there is no overlap. Some tissues are characterized by a marked predominance of one type. For example, type 2, 9, 10, and 11 in cartilage and type 1 collagen in bone. It is likely that each of these collagen types is large and is responsible for the functional and the morphologic properties of each connective tissue, although it has not yet been possible to relate function to a particular chemical modification. So as it is visible in this slide, so there are specifically several steps. So as it is visible that there are propeptides and then there is a conversions in the endoplasmic reticulum. It converts into procollagen. Further, it moves into the Golgi complex for the lateral association. Thereby, it leads to the propeptide cleavage and thereafter it results into collagen fibrils, which finally it assembles and cross-links. So, collagen is a highly abundant and varied protein with more than 14 types being discovered. All variations are formed from tropocollagen molecules which are inelastic. The tropocollagen molecule is formed from three peptide chains. Each chain is formed from one-third of glycine, one-third of proline and hydroxyproline and one-third other amino acids. The different collagen types are formed by variations in the sequence and amount of amino acid found in the alpha chain. The three chains are principally stabilized by the van der Waals bonds and some occasional covalent bonds, forming a right-handed helix. The helix is around 300 nanometer in length and 1.5 nanometer in diameter, with a screw pitch of 0.27 nanometer. The tropocollagen molecules are arranged in a quarter staggered array with a 26.5 nanometer overlap and a 37.5 nanometer gap between each end of the molecule. A sequence of tropocollagen molecule is the microfibril. These microfibrils are organized in parallel to form a collagen fibril with a 50 nanometer diameter. Further, the collagen fibrils then twist together to form a single collagen strand, and it is the cross-linking between the molecules at each structural level that gives collagen its high tensile strength. Further, as visible, it shows the chart of some of the important types of collagen and their properties, which includes how, like in which polymerized form they are formed and in which tissues they are distributed. So talking about the fibrillar type, so it comprises of type 1, 2, 3, 5 and 11. So collagen type 1, it is in the form of fibril as it is in the group of the fibrillar group, and it is distributed in the bones, skin, tendons, ligaments, cornea and internal organs. Further, the collagen type 2, it is also in the form of fibril and it is found in the cartilage, in vertebral disc, notochord, vitreous humor of the eye. Similarly, type 3, it is found in the skin, blood vessels, internal organs. And type 5, type 11, they are found as for type 1 and for type 2 respectively. Talking about the fibril associated, it contains of type 9 and type 12. So the type 9, it is found in the polymerized form where it is in the form of the lateral association with type 2 fibrils and it is distributed in the cartilage. Further, type 12, it is found as in the lateral association with some type 1 fibrils and it is found in the tendons, ligaments and some other tissues. Further, talking about the network forming type, it is found in the type of the type 4 and the type 8 and it is found in the form of the sheet-like network and the anchoring fibrils respectively. Further, coming to the type of the tissue distribution, it is found in the basal lamina and also beneath the stratified squamous epithelia in case of type 7. Further, the other two types are the transmembrane and other types, and they are generally found in the hemidesmosomes and the basal lamina around the blood vessels. Further, moving towards the details, so the different types of collagen can co-assemble to form large fibers. Like the type 6 and the type 1 in tendons, they form in direction of the stress. Further, type 2 and type 9 are oriented randomly and are in cartilage for strength and compressibility. So there can be various types. For example, the type 1, which is the chief component as it can be shown in the image. So the type 1 is the chief component of the tendons, ligaments and bones. Further, the type 2, it represents more than 50% of the protein in cartilage and it is also used to build the notochord of vertebrate embryos. Further, type 3, it strengthens the walls of the hollow structures like arteries, the intestine and the uterus. Then type 4, it forms the basal lamina of epithelia, which is the basal lamina is often known as the basement membrane, but it is not related to lipid bilayer membranes. So a meshwork of type 4 collagen, it provides the filter for the blood capillaries and the glomeruli of the kidneys. Talking about the osteogenesis imperfecta, so type 2 collagen is found predominantly but not exclusively in the hyaline cartilage, where it accounts for 90% of the total collagen. Further, type 2 exists in two splice variants, which is the 2A and the 2B. The dominant form is the 2B, which is found in mature cartilage, and exon 2 is spliced out. In 2A, a transient embryonic form found in prechondrogenic mesenchyme, perichondrium, and vertebra. This domain is retained. And further, the collagen 2 interactions, talking about those interactions. So the collagen 1 domain, it interacts with the integrin receptors, principally the alpha 2 beta 1 and the non-integrin binding protein anchorin C2. The fibrillar form of type 2 collagen is cross-linked to type 9 collagen via lysine, hydroxylysine residues in the N and C telepeptides of the type 2 collagen and also in the helical called 2 domain of type 9. The fibrillar form of type 2 collagen also interacts with the protein cores of the fibromodulin and decorin. Talking about the interacting proteins, so decorin, fibromodulin, lumican, chondroadenine, comb, fibroactin, and aggregan, which are K and S rich regions. Further, talking about the laminins, so these are the multi-adhesive matrix proteins and their major function is the organization of basement membrane, and they have binding sites for integrin receptors which are really important in the embryonic development and also in the tissue remodeling. Coming to the structure, so they are really tightly associated with entactin or nidogen, which is a 150 kilodalton sulfated glycoprotein, which also binds to type 4 collagen. As a result of these several multiple interactions, laminin, intactin, type 4 collagen, and perclan form cross-linked networks in the basal lamina. So laminins can be considered, and laminins are the extracellular matrix proteins that majorly consist of the alpha, beta and gamma chains with molecular masses in the range of 140 to 400 kilodalton. Chain association occurs through a large triple alpha helical coiled coil domain towards the C terminus of each chain. 8 genetically distinct laminin chains, which is alpha 1, 2, 3, beta 1, beta 2, beta 3, gamma 1 and gamma 2, and 7 different assembly forms, which are the laminins 1 to 7, are known so far. The most extensively characterized laminin 1, which is the alpha 1, beta 1 and gamma 1, it shows calcium dependent self-assembly and heterotypic binding to the perlican, nitrogen, fibrillin 1 and other matrix components. This binding indicates a crucial role in the supramolecular organization of basement membranes. Further, laminins also possess binding sites for at least 6 different integrin receptors and are thus involved in many cell matrix interactions. Such interactions have been shown to be important during the embryonic development, as I already mentioned, and also for tissue homeostasis and remodeling. So the globular Lg domains at the C terminus, it mediates the calcium dependent binding to specific carbohydrates on certain cell surface molecules such as the synticon and the dystroglycan. So the Lg domains can mediate binding to steroids and proteins as well. Further, the transmembrane protein dystroglycan is a highly glycosylated central element of the dystrophin-associated glycoprotein complex, which is involved in the pathogenesis of many forms of transmembrane protein. Dystroglycan is a highly glycosylated central element of the dystrophin-associated, and it is involved in muscular atrophy and muscular dystrophy pathogenesis. Dystroglycan is a receptor for multiple extracellular matrix molecules such as that of laminin, agrine and perlican, and also plays a role in the linking the ECM to the actin cytoskeleton. However, these interactions are regulated and their basic cellular functions are poorly understood. Drosophila dystroglycan is linking the ECM to the actin cytoskeleton. However, how these interactions are regulated and the basic cellular functions are performed, these are poorly understood. Drosophila dystroglycan is required for the cell autonomously for cellular polypolarity in two different types. The epithelial cells, which is for the apicobasal polarity, and the oocyte, which is for the anteroposterior polarity. So loss of dystroglycan function in follicle and disc epithelia results in expansion of apical markers to the basal side of the cells, and overexpression results in reduced apical localization of the same markers. Further, we'll discuss about the adhesions. So, the adhesions between the non-epithelial cells and the extracellular matrix are short and long-term adhesions that generally helps in the motility. So, it can be the focal adhesions, the focal contacts, the focal complexes, the 3D adhesions, fibrillar adhesions, and the porosomes. So this table, which is visible right now in front of you, it shows the selected and several types of vertebrate integrins that are present. So this chart shows the several subunit composition and the primary cellular distribution along with its ligands. So talking about the first one, which is the alpha 1 beta 1. So basically it is found in many types, and it the major ligands are mainly the collagens and also laminins. Further, the second subunit composition is alpha 2 beta 1, which is again found in many types, and the major ligands are mainly collagens and also laminins. Thereafter, another subunit, which is alpha 4 beta 1. So it is found majorly in the hematopoietic stem cells and hematopoietic cells, and the major ligands lies the fibronectin and the VCAM1. So moving on to other types, which is the alpha 5 beta 1, which is the found in the fibroblast, and the major ligands are fibronectin. So like this, it shows the several vertebrate integrins and how they play a major role and major function in proper functioning of the human body.